Dr. Sage here. Today we're going to discuss Chapter 5. In Chapter 5 we learn about the macromolecules. These are the large biological molecules that are required for life. All living things are made up of four classes of large biological molecules. Those are called carbohydrates, lipids, proteins, and nucleic acids. Macromolecules are very large molecules composed of thousands of atoms connected together by covalent bonds. What we're going to see throughout this lecture is that the structure of the molecule is what determines the function of the molecule. Alright, so for the format of this chapter, chapter 5.1, the first section, is essentially like a very brief introduction to macromolecules, like in general what they are. Then we're going to discuss each of the different classes of macromolecules separately. So 5.2 we'll talk about the carbohydrates, 5.3 we'll discuss the lipids, 5.4 we'll talk about proteins, and then 5.5 we'll talk about the nucleic acids, DNA and RNA. So, as your general introduction to macromolecules, a polymer is a long molecule consisting of many similar building blocks. These small similar building block molecules are called monomers. An analogy for that. Let's say that you're playing with Legos. So as you're playing with Legos, each of the Lego pieces, they're all very similar to each other. Like they might be the same size and the same shape, but they're not exactly the same. Like they come in different colors. Now you take those Lego pieces and you put them together to build something, like let's say a Lego castle. Alright, so with that analogy, the Lego pieces, the small similar building blocks, those would be equivalent to the monomers. That large structure you built out of these small similar building blocks, like the Lego castle, that would be equivalent to the polymer. So monomers are the small pieces used to build the polymers. Now, out of the four large macromolecules, technically 3 of them are polymers and 1 is not a polymer. So carbohydrates, proteins, and nucleic acids are polymers. Technically, lipids are not a polymer. And that will make more sense whenever we learn about each of the different classes of macromolecules. Okay, so let's say you're playing with Legos. Okay, if you're playing with Legos, you want the ability to take those Lego pieces and put them together to build something, like a Lego castle. But let's say you get done playing with that Lego castle, like you're bored playing with the Lego castle. Well, you're not going to take the whole castle and just throw the whole thing away. What you're going to do is you're going to take that Lego castle and break it apart into smaller pieces, the Legos, so you can reuse them to build something else, like let's say a space shuttle. Well, you want to be able to do the same thing with your molecules. You want to be able to take the monomers and build polymers, but then when you're done with that polymer, let's say a protein, you don't want to just throw it away. You want to break it back down into the monomers so you can reuse them to build a different protein. Now, if you're not playing with Legos, if you're talking about molecules, then to build them and break them down, those are going to be chemical reactions, which are called a dehydration reaction and a hydrolysis reaction. So a dehydration reaction occurs when 2 monomers bond together through the loss of a water molecule. The opposite of that would be hydrolysis. Polymers are disassembled to monomers by hydrolysis, a reaction that's essentially the reverse of the dehydration reaction. Alright, so let me explain those in a figure. So let's say you want to build some polymer. And it doesn't actually matter which polymer it is. You build them all in the same way. So whether you're building a protein, or you're building a complex carbohydrate, or you're building a DNA molecule or an RNA molecule, you build them all in the same way. The pieces are different, like the Lego pieces, the monomers are different between like proteins and DNA, but how you build them is the same. So let's say you started to build your polymer, and you want to keep building it to make a bigger polymer. What you're going to do is you're going to take your next monomer, your next Lego piece, and what you want to do is you want to form a covalent bond between them. So you would grow your polymer by 1 more monomer. You had 3 monomers that were making up this polymer, you now have 4. So you want to build this covalent bond here. Well, in order to build that covalent bond, you have to take this H and this OH and remove them to replace them with this covalent bond. So you're removing an H and an OH, or you're removing 2 H's and an O, or you're removing a water molecule to form this covalent bond. And that's actually where the name for this reaction comes from. This is called a dehydration reaction because you're dehydrating or you're losing a water molecule to form that covalent bond. Okay, so that's a dehydration reaction, and that's how you build all of your polymers from their monomers. Now, you want to be able to do the opposite. You want to be able to take that Lego castle, break it down into Lego pieces. How you do that is basically the opposite chemical reaction. You want to break this covalent bond here. Okay, well, in order to break that covalent bond, you have to add back the H and the OH you removed, or you have to add back a water molecule. Okay, and this is how you break your polymers into monomers. You just keep breaking one at a time these covalent bonds to break this polymer down into monomers. Now, this is called a hydrolysis reaction, and again, the name is telling you what's happening. Hydro is referring to water, and lysis means break. You're breaking the water in half to put half of it here and half of it here, or if it helps you to think about it, you're breaking this covalent bond by using this water molecule. Okay, so that's how you build and you break down polymers. Okay, so that was section 5.1. Now, the rest of the sections are not going to be that short. That was just a brief introduction. Basically, just talking about what are macromolecules, what's a polymer, what's a monomer. Now, what we're going to do is we're going to start going through each of the macromolecules. The first one we're going to talk about is the carbohydrates. Carbohydrates include sugar molecules and the polymers of sugar molecules. The simplest carbohydrates are called monosaccharide or single sugar molecules. Okay, this prefix, mono, that means one, and saccharide means sugar. So this is a single sugar molecule. Those are going to be the monomers that are used for the carbohydrates, like the Lego pieces, the monomers, they're called monosaccharides. Now, you take those monosaccharides and you string them together. You take your Lego pieces and you build like a Lego castle out of them. That makes the polymer, which for the carbohydrates, those are called polysaccharides. Poly means many, saccharide means sugar. Okay, so the monomers are called monosaccharides. The polymers are called polysaccharides for the carbohydrates, the sugar molecules. Now, just like there are different Lego pieces, there are different monosaccharides. And just like all the Lego pieces are similar to each other but not exactly the same, all the monosaccharides are similar to each other but not exactly the same. So the monosaccharides all have the same basic chemical formula, CH2O. So you could have a monosaccharide that would be like C3H6O3. So equal numbers of carbon and oxygen and twice as many hydrogens. Or you could have a monosaccharide that's C6H12O6. Okay, they all follow the same basic formula. Equal numbers of carbon and oxygen and twice as many hydrogens. Now, there are different monosaccharides, and they can be classified based upon the location of the carbonyl group as aldose or ketose. Okay, pretend I did not say that. If you want to learn about that, take chemistry. Or based upon the number of carbons in the carbon skeleton. So, for example, these are all monosaccharides. And you get a monosaccharides built out of different numbers of carbon. Okay, like this one here is built out of 3 carbons. So since you have 3 carbons, you will also have 3 oxygens, and then you have twice as many, or 6 hydrogens. Okay, so that's an example of a monosaccharide. Or you could have monosaccharides built out of 5 carbons, or monosaccharides built out of 6 carbons. Okay, those are all examples of monosaccharide single sugar molecules. The monosaccharides are listed on this figure. Actually, most of them we're going to ignore for this course. The ones we're going to focus on in this figure are actually just these two, ribose and deoxyribose. And we're actually not going to focus on them yet. We're going to talk about them when we get to the last section of this chapter, which is a section that discusses nucleic acids, which is DNA and RNA. Well, it turns out that DNA and RNA are built out of things, and one of the things they're built out of is sugar molecules. Okay, well, these are the sugar molecules inside DNA and RNA. And in fact, the names for DNA and RNA are actually telling you that. What DNA actually stands for is deoxyribonucleic acid. Okay, well, deoxyribonucleic acid uses deoxyribose as its sugar molecule. This is a sugar inside DNA. RNA actually stands for ribonucleic acid. Well, ribonucleic acid uses ribose as its sugar molecule. This is a sugar inside RNA. And these two sugar molecules, when you look at them, they're very similar to each other. In fact, basically the only difference is this one has an oxygen here and this one does not have an oxygen there. Okay, so in other words, deoxyribose is very similar to ribose. In fact, deoxyribose is ribose that's been deoxygenated. It's lost an oxygen. Okay, so again, the name is telling you what's going on with that molecule. Okay, but these two we're going to come back to discuss in the last section of this chapter when we talk about DNA and RNA. So the monosaccharide we're going to focus on in the carbohydrate section is actually glucose. Okay, glucose is a 6-carbon sugar, and it comes in a couple of different forms. Like it could be in a straight chain while the carbons are in a straight line, or it can be a ring form like this here. Okay, these are both glucose. It's just whether this carbon and this oxygen will form a covalent bond with each other to form a ring instead of a straight line. Okay, but they're both glucose molecules. More often, this is how you find it inside your cells, in this ring form. In fact, the rest of the time, it's going to be diagrammed as a little hexagon shape like this. So this is a monosaccharide we're going to focus on when we're talking about carbohydrates. So why we're going to focus on glucose in particular? Because it turns out that your cells need energy for everything they do. Moving a muscle takes energy. Thinking actually takes a lot of energy. Building molecules like DNA or proteins takes energy. Where you get your energy from is the food you eat. Okay, well, your cell's preferred source of energy, the energy your cells actually like the most, is glucose, which is pure sugar. Okay, so since it's your cell's preferred source of energy, that's the monosaccharide we're going to focus on in Bio 1. Okay, but you have different monosaccharides, and those are some examples of them, and like I said, we're going to focus for this section at least on glucose. So mono means one, so a monosaccharide is a single sugar molecule. Next, what you can have is disaccharides. Okay, this prefix di means two, so that's two monosaccharides joined together by a covalent bond. Okay, they are just joined together by a covalent bond because they like to be a pain in science. They give it a special name. It's called a glycosidic linkage, but it's just a covalent bond that joins them together. Now, because you can have different monosaccharides, different Lego pieces, what that means is you can also build different disaccharides. So some examples, okay, ones you might have interacted with before. So for example, table sugar, that's sucrose. Like that's the sugar you add to your coffee, for example. Okay, that's a disaccharide. It's two monosaccharides joined together. This is made out of two different types of monosaccharides, made out of glucose and fructose, which are two different monosaccharides. Another example, another one you might have heard of before, because you might have heard of people who are lactose intolerant. Well, lactose is the sugar that's inside milk. Lactose is a disaccharide made out of galactose and glucose. Okay, so that's another disaccharide you might have heard of before. Okay, so you don't need to memorize these. The point in showing you these is just the general point that since it's more than one monosaccharide, you can make more than one type of disaccharide. So you have monosaccharides, single sugar molecules, disaccharides, two monosaccharides linked together. Technically, next you would have oligosaccharides. That's where you have like 8 or 16 monosaccharides joined together. We might talk about that in a later chapter. But then after that, what you have is you have the polysaccharides. Okay, these are the polymers. Remember, poly means many, so this is many sugar molecules joined together. Okay, so monosaccharides are the monomers, polysaccharides are the polymers, or like the Lego castle. Now, there are also different types of polysaccharides. The three main polysaccharides we're going to focus on are called starch, glycogen, and cellulose. Okay, so these are three different types of polysaccharides. But it turns out all three of these polysaccharides, they are all built out of the same type of monosaccharide. All three of these are just long chains of glucose. They're all built out of glucose. So if they're all just long chains of glucose, then what's different about them? Okay, well, let's talk about the first two. Okay, so starch and glycogen. Okay, starch you find inside plant cells, whereas glycogen you find inside animal cells. Okay, they're both long chains of glucose, but starch is in plants, glycogen is in animals. Now, the purpose of starch and glycogen, the purpose of both of them is to store glucose for later energy use. Remember, I said your cells need energy, and their preferred source of energy is glucose. Well, let's say you took in more glucose than you need. Well, your body's not going to throw away that glucose. It's going to store it. And one way it can store it is in long chains, like linking the glucose molecules together. Okay, and in animals, that's glycogen, like you have glycogen inside your cells. And in plants, that's called starch. Okay, so these are long chains of glucose for later energy use. All right, so that's starch and glycogen. What about cellulose? Okay, well, cellulose is in plants, just like starch is in plants. Cellulose is long chains of glucose, just like all three of these are long chains of glucose. Okay, so what's different about cellulose? Well, the cellulose are the long chains of glucose not for later energy use. Instead, what they are is they're a building material, like they form structures inside plant cells. So let me give you some examples of that, a little bit better descriptions. Differences between them. Cellulose, that's a long chain of glucose. So each of these little hexagons that represents a glucose molecule. So cellulose is a long straight chain of glucose. Starch and glycogen are also chains of glucose, but instead of being a long straight chain, it's branched chains of glucose. Or maybe a nicer way is visualizing it. Each of these green hexagons represents a glucose molecule. So in starch and in glycogen, starch in plant cells, glycogen in animal cells, they're long chains of glucose, but they're not just straight chains of glucose, they're branched chains of glucose, like branches on a tree. Okay, both of these are storing the glucose for later energy use. So let's say you need some energy. What you're going to do is you're going to take this glucose molecule and break it off of the glycogen and use that glucose for energy. Okay, so starch is in plants, glycogen in animals, both long branched chains of glucose for later energy use. Cellulose is in plants, but it's not a long branch chain, it's a long straight chain of glucose. Now, when you get long straight chains, what happens is you can pack them very tightly together, like you see here. Now, when they're packed very tightly together, what happens is it forms a stiff, rigid structure. And plants use that as a building material. For example, let's say you take a leaf of a plant, okay, and let's say you put it underneath a microscope. In this case, this is what you're going to see. Kind of what's inside this yellow highlighted region, that'd be one plant cell. And this yellow highlighting you see right here, what that is the plant cell wall. Now, if you take that plant cell wall and you put it under a very powerful microscope, like an electron microscope, it's going to look like it's made up of a bunch of fibers, okay? Because it is made up of a bunch of fibers, okay? If you could zoom in or magnify more, you're going to see it's made up of cellulose fibers, long straight chains of glucose. Okay, and that plant cell wall gives the plant cell its shape and supports the plant cell and protects the plant cell. We'll learn about it in a later chapter, but it's made out of cellulose. Okay, so that's starch, glycogen, and cellulose, three types of polysaccharides. They're all made out of glucose. Now, there are other types of polysaccharides. Just as one more example, here's a polysaccharide that's not built out of just glucose. Okay, chitin. Chitin is a polysaccharide that's made out of other types of monosaccharides, like glucosamine, for example. Now, chitin is used as a building material, just like cellulose is used as a building material. Where do you find chitin? Inside the hard shells of beetles, you find chitin. So technically, beetle shells are made out of sugar. I'm not saying go eat a beetle, but they are built out of sugar. Okay, so that's another example of a polysaccharide, okay, which that is our discussion on carbohydrates. One of the types of macromolecules, the large biological molecules required for life. Okay, in section 5.3, we're going to discuss lipids. So lipids are the one class of large biological molecules that technically is not a polymer. Okay, and there are different types of lipids, but the main unifying feature of lipids is they have little or no affinity for water. In other words, lipids don't like water. In other words, lipids are hydrophobic. Okay, now the reason that lipids tend to be hydrophobic is because they consist mostly of hydrocarbons. Alright, so what a hydrocarbon is a molecule that's almost entirely made up of hydrogen and carbon. Now recall from the chemistry lecture, the covalent bonds between hydrogen and carbon are non-polar covalent bonds. So there's an equal sharing of electrons, so there's no partial negative or partial positive electrical charges, so there's nothing for water to be attracted to. So because of that, lipids are hydrophobic. Now, there are different types of lipids. The ones we're going to focus on in this section of the chapter are fat, phospholipids, and steroids. We're going to talk about these three types of lipids. So the first type of lipid we're going to talk about is fat. Fat is made up of two smaller types of molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon, and a fatty acid consists of a carboxyl group attached to a long carbon skeleton. Okay, and those words probably just made no sense to you. So what that was actually saying is this is a fat molecule. Okay, a fat molecule is made up of two types of molecules. Okay, in this kind of beige shaded box here, that's called glycerol. And then 3 molecules of fatty acids. Okay, so fat is made out of glycerol and fatty acids. So it has these 3 fatty acid, what we're going to call fatty acid tails. Okay, so this is fat. Now, it's technically not called fat. Okay, its technical name is, this is actually called a triglyceride. So fat is the same thing as a triglyceride, and that's what this is, fat or a triglyceride, which is a type of lipid. You've probably heard that term before, triglyceride, like you might have had blood work done, and they might have discussed your triglyceride levels. Okay, that was talking about the different types of fat in your blood. This is a fat molecule, a triglyceride, which is a type of lipid. Now, there are different types of fat molecules. One way you can have different types of fat molecules is the fatty acid tails can be of different lengths. Like you can see, this fatty acid tail is longer than this fatty acid tail. So that's one way you can have different types of fat molecules. Another way you can have different types of fat molecules is something you've probably heard of before in terms of your nutrition, because you've probably heard of saturated fat versus unsaturated fat. Okay, well today we're going to learn what that term actually means, saturated fat versus unsaturated fat. Okay, so for example, this fat molecule here, the way it's currently drawn, that is a saturated fat. A saturated fat is a molecule where you have no double bonds between any of the carbon atoms. So if you notice, none of these carbons have double bonds between them, so that's going to be a saturated fat. In contrast, an unsaturated fat has some double bonds between the carbon atoms. So let's say I draw a double bond here between these two carbons. Now, the way I drew this double bond is actually a problem. If you refer back to the chemistry chapter, what I taught you is that a carbon always has to have 4 covalent bonds. Well, this carbon right here has 1, 2, 3, 4, 5 bonds. Okay, that's not possible. So in order to form this double covalent bond, what has to happen is this carbon has to lose this hydrogen atom. Okay, so it doesn't have that hydrogen in there. On the other end of the bond, this carbon has 1, 2, 3, 4, 5 bonds. Again, that's not possible. So to form that double bond, you also have to lose this hydrogen atom. So this is an unsaturated fat. The reason it's an unsaturated fat is because it has some double bonds between the carbons, and because of that, it is not saturated, not filled up with as many hydrogens as it could have. Okay, because it's missing a couple of hydrogens here. So it's not saturated, not filled up with as many hydrogens as possible, so it's called an unsaturated fat. In contrast, this fat molecule that has no double bonds between the carbon, it is saturated or filled up with as many hydrogens as possible. So that's why it's called a saturated fat, because it's filled up with as many hydrogen atoms as possible. So that's a saturated fat. Now, as I mentioned, fat or a triglyceride is a type of lipid. Remember, one of the main defining features of lipids is they're hydrophobic. They hate water. The reason they tend to be hydrophobic is because they're mostly made out of hydrocarbons. Okay, it's like, for example, these fatty acid tails here. This is entirely just carbon and hydrogen, what I highlighted here. So since this molecule is almost entirely just carbon and hydrogen, this is non-polar covalent bonds. There's no partial negative or partial positive electrical charges, nothing for water to be attracted to, so it's going to be hydrophobic. It's not going to like mixing with water. So saturated fatty acids have no double bonds between the carbons. It's saturated, filled up with as many hydrogens as possible. Unsaturated fatty acids have some double bonds between the carbons, so they are not saturated, not filled up with as many hydrogens as possible. Now there's also different types of unsaturated fat. And again, you might have heard of this in terms of your nutrition. On your food label, you might have read about monounsaturated fat, polyunsaturated fat. Okay, remember this prefix mono means one. So a monounsaturated fat has one double bond. Poly means many. So a polyunsaturated fat has more than one double bond. Okay, so why do we care if the fatty acids in the fat molecules are saturated or unsaturated. The reason we care is because it changes the behavior of the molecule. So for example, this is a fatty acid. Remember, a fat molecule has 3 fatty acids. Okay, and this fatty acid here is a saturated fatty acid. There's no double bonds between any of these carbons, so it's saturated or filled up with as many hydrogens as possible. Now, when you have a saturated fatty acid, what happens in reality, in 3-dimensional space, the molecule forms a long straight chain. When you have molecules that form long straight chains, what happens is they can pack very tightly together, like you can see here. That's a saturated fat. Now, in contrast, if you have an unsaturated fat, it's going to have some double bonds between the carbons, so it's not going to be saturated or filled up with as many hydrogens as possible. And when you form these double bonds, what it does is it forms kinks in the chain. So these unsaturated fatty acids, because they're kinked, they're not a long straight chain, they cannot pack as tightly together. Like, compare these 4 unsaturated fatty acids to these 4 saturated fatty acids. These can pack much more tightly together. These cannot pack as tightly together. Okay, and that changes the behavior of the molecule. For example, animal fats tend to be saturated. So because of that, they're packed tightly together. And because of that, they tend to be solids at room temperature. Remember from the last lecture, my sad little states of matter dance, and what I told you is that in a solid, the molecules tend to be very close together, just vibrating next to each other. Whereas in a liquid, the molecules are farther apart and they're moving around each other. So this saturated fat, these molecules are packed very tightly together. Just like in a solid, molecules are packed very tightly together. In this unsaturated fat, these molecules are not packed as tightly together, they're spaced out more. Just like in a liquid, the molecules are farther away from each other. So animal fats are saturated, they're packed together, they tend to be solids. Plant oils tend to be unsaturated. The kinks in the chain prevent them from packing tightly together, so they tend to be liquids at room temperature. All right, so for example, butter versus olive oil. Okay, these are both the same type of molecule. Butter is a triglyceride. It is fat. Olive oil is a triglyceride. It is fat. So what's the difference between them? Butter is made out of saturated fatty acids. Because they're saturated fatty acids, no double bonds, as many hydrogens as possible, and they form long straight chains, so they tend to pack very tightly together, so it tends to be a solid at room temperature. Like if you take your butter out of your fridge, leave it out at room temperature, it's a solid. It's a soft solid, but it's a solid. In contrast, olive oil, okay, it's also fat. It's also a triglyceride, but it's an unsaturated fatty acid. It has some double bonds in the carbons. Okay, because of that, it does not have as many hydrogens as possible, and it forms kinks in the chain. Okay, because it has kinks in the chain, it cannot pack as tightly together, so it tends to be a liquid at room temperature. So that's the difference between butter and olive oil. They're both fat, they're both triglyceride. Okay, the difference is one is saturated fat, the other is unsaturated fat. A diet rich in saturated fats may contribute to cardiovascular disease through plaque deposits. In other words, most people are pretty aware of this nowadays. Generally, most people feel that saturated fat is less healthy for you, unsaturated fat is more healthy for you. Okay, so what do? We as humans, I guess because we're really dumb, what we do is we take something that's healthier for us, like olive oil, and we essentially turn it into something that's less healthy for us. What we do is we take this unsaturated fat and we turn it into a saturated fat. Okay, you've probably heard of that because you probably on your food labels heard about hydrogenated vegetable oil. What this is this is where they take vegetable oil, remember this is a healthier type of fat, the unsaturated fat, and they turn it into saturated fat by adding the hydrogens back. Why do we do this? Because we're dumb. But also, I'm sure it tastes a lot better, and that's probably the main reason. But when we do this, We also can create trans double bonds. Take chemistry or ask me later if you want to know exactly what a trans double bond is. But this creates trans fats, which are the most unhealthy fat you can possibly eat. The things that are most likely to cause cardiovascular disease. Trans fats are made by us. Okay, we take something that's healthy, vegetable oil, and we turn it into a trans fat. Why? Again, because I said we're stupid. But joking aside, the real reason is at first I assume it tastes really good, and then also trans fat has a much longer shelf life. It lasts longer than unsaturated fat. But these are so bad for you that a lot of cities have banned their use in restaurants. Or if you go to the grocery store and you're reading your food labels, a lot of times it will say it contains no trans fats. You know, at the very least, it does not contain the worst possible type of fat for you. Now, some unsaturated fatty acids, remember those are the healthier ones, are not made by the human body, and we actually need them. So since we cannot make them ourselves, we have to get them by our diet, by eating them. And again, you might have heard of these before because you might have heard of omega-3s versus omega-6s. Omega-3s are required for normal growth, and they're thought to provide protection against cardiovascular disease. So these don't hurt your heart, these help your heart. These are in things like walnuts or fish oil. And that's why you might hear about people taking fish oil supplements, because it contains this healthy type of fat. All right, so what do we use fat for? The main purpose of fat is to store energy. All right, we will learn more details about this in a later chapter, but it turns out there's energy stored in chemical bonds. Well, this fat molecule has a lot of chemical bonds in the backbones of these fatty acids. So this fat molecule is storing a lot of energy, and that's the main thing we use fat molecules for, is to store energy. Now, humans and other mammals store fat in their adipose tissue or adipose cells. Besides storing energy for later energy use, adipose tissue also cushions vital organs and helps to insulate our body. It helps it to make it easier for us to maintain our body temperature. Okay, so that is fat or triglyceride, which is your first type of lipid. Now let me jump back to a second to the structure of the fat molecule, or triglyceride, which is one type of lipid. Remember, fat or triglyceride is made out of glycerol and then 3 fatty acid tails. Okay, the reason I'm pointing this out is because the next type of lipid we're going to talk about is called a phospholipid, and its structure is kind of similar. It's built out of glycerol and 2 fatty acid tails. In place of this third tail, we have something else. Okay, so this is fat or triglyceride, one type of lipid. The next type of lipid we're going to talk about is called a phospholipid, which is made out of glycerol and 2 fatty acid tails. In place of the third tail, you have a phosphate group. And don't worry about this for this course. There's a phosphate group. Okay, so that's a phospholipid. Now, a phospholipid is kind of a unique molecule because it's amphipathic, which amphipathic is a chemistry term that means it has 2 opposite chemical properties at the same time. So, for example, these tails here, they are hydrophobic. They hate water. But what we're going to call up here the head of the molecule, that's hydrophilic. It loves water. So we have a molecule where part of it is hydrophobic, water hating, part of it is hydrophilic, water loving. The rest of the time we're going to diagram it like this just because it's easier to visualize. You have the hydrophobic, water hating tails, hydrophilic, water loving head of the molecule. Now, if you take phospholipids and just dump them in water and don't do anything else, just drop them in water, all on their own, they form this shape, which is called a phospholipid bilayer. The reason it's called a phospholipid bilayer is because it's 2 layers of phospholipids. There's phospholipids here and phospholipids here. Okay, they form this shape all on their own without you having to do anything. If you just dump them in water, they'll assemble into this shape. Now, why do they do that? Because they're amphipathic. The water-hating tails, they don't want to be near the water. So what the water-hating tails do is they all clump together to try to get away from the water. In contrast, the water-loving heads of the molecules, they love water. They're hydrophilic, so they all want to face out towards the water. This molecule, a phospholipid, forms this shape, which is called a phospholipid bilayer, all on its own. Okay, why do we care that phospholipids form a phospholipid bilayer? Because we're studying biology, the study of living things. All living things are built out of cells. if you look at a cell, like when people think about cells, they think of like a bag with like a bunch of stuff inside it. You know, it's a very rough description of a cell. We'll learn obviously in later details what a cell really is. But if you think of a cell as a bag with a bunch of stuff inside it, the bag itself, like the Ziploc bag with the stuff inside it, that bag, that's called the plasma membrane, that is a phospholipid bilayer. So it turns out all of your cells are built out of a phospholipid bilayer. And all living things are built out of cells, so that's why phospholipids are very important in biology. In fact, we're going to spend an entire chapter talking about membranes like this. So obviously, there's a lot more details than just phospholipids, but we'll get to that in a later chapter. Okay, so phospholipids is very important because they form the phospholipid bilayers. So that's your second type of lipid. The third type of lipid we're going to discuss in this chapter is steroids. Okay, so a steroid is not one thing, it's a category. Okay, steroids are lipids, so they're hydrophobic, and they're characterized by having a chemical structure that's built out of 4 fused chemical rings. So all steroids have those 4 fused chemical rings. That's what makes it a steroid. Now, some things are steroids that you might think of, all right, yeah, that logically makes sense that that'd be a steroid. So for example, testosterone is a steroid. But there are other things you might not think of as being steroids that are steroids. So for example, cholesterol is a steroid. Vitamin D2 is a steroid. So what makes these things steroids is they all have these 4 fused chemical rings. That's what makes it a steroid. That's what defines it as being a steroid. But in your body, testosterone does not do the same thing as cholesterol. What makes testosterone different from cholesterol is what's attached to those 4 fused chemical rings. Now, we're not going to go into details or learn what is cholesterol doing, what is testosterone doing inside your body. After this course, take anatomy and physiology, and you'll learn more details about that. So for this class, what you should know about steroids is they're a type of lipid. They are hydrophobic, water-hating, and they're defined by having these 4 fused chemical rings. That's the level of details we're going to worry about for steroids. Okay, so those are lipids. Lipids are fat, which is triglyceride, phospholipids and steroids. Those are lipids we're going to discuss. They're hydrophobic. They don't like water. Okay, they are one of the 4 macromolecules that is not a polymer. Because remember, a polymer is a long chain of monomers linked together by covalent bonds. These are not monomers linked together by covalent bonds. So lipids are not polymers. That's the 1 out of the 4 is not a polymer. So far we've talked about 5.1, your general introduction, 5.2, which is carbohydrates, and 5.3, which is lipids. 5.4 that we're going to discuss, that would be the next thing to discuss. That's going to talk about the proteins. And then 5.5 is going to talk about the nucleic acids, DNA and RNA. What I'm going to do is I'm going to break this up into 2 separate videos. So I'm going to end this video here. So this is the first half of Chapter 5. The next video, we're going to discuss the second half of Chapter 5, which is proteins and nucleic acids. So until then, this has been Dr. Sage.